with like proper soap, or like 60% of some that can actually be that can clean your hand properly, and if you're not doing that, you can be exposed to the viruses. All right, so it's very, very important that you keep washing your hands and you avoid touching as much as you can to your, especially nasal areas or your mouth or your eyes, so that way you do not get exposed to these viruses. All right, so that's the reason why it's important to be your hygiene. It's important to have like this respiratory hygiene, plus like your hand hygiene or your body hygiene too. All right, and other thing is that being very, very cautious. Okay, and that also, like sometimes we do not know any sort of like vertical transmission. What does that mean? Meaning like we don't know for sure, like either the mother can give it to, like if the mother is pregnant, whether and she's infected the virus, whether she can actually give it to the child or not. We do not know that. And other one is like the fecal route. We gotta mention about that too, right? The fecal route, there's still investigations going on, but we do not know for sure whether there's actually the virus that can actually transmit it to the fecal route for so far. Okay, so far, but these are the common things that you can transmit it to viruses. And remember, like when you sneeze at someone, right, like you are sneezing, like, to be honest with you, if you want to look at it number wise, you are sneezing millions and millions of viruses, about like millions of viruses to someone whenever you sneeze. So that's why it's really important that whenever you sneeze, you don't sneeze on your hands, or like there's a proper way you sneeze. You sneeze on here, right here, right? So, and making sure that you're actually cleaning your hands properly. Okay, and you took like extreme precautions for this. Okay, but you do also, one more thing that I do have to mention is that like this word becomes so famous here, like self-quarantine. I know it's important that you're quarantining yourself and maintaining the physical distance from not only your friends or your loved ones, but also it's important to, like if you're one of those people that you have been going out to work, it's also important that you physically kept distant from your families as well, because of that way you do not give, especially to your elderly people, if they already have a preconditioned disease. Okay, that's also, keep that in mind. And also one more thing is that a time like this, whenever we are like self-quarantining ourselves, it's important that we are going to take care of ourselves too, right? Because like places like here, where I live, it's New England, right? So I'm always like sort of like not really exposed to sunlight, and because of what happens is that my vitamin D can go down, right? So it's important for me to actually it's important for me to sort of like, if I'm just quarantining myself, and if I'm not going out, if I'm not getting sunlight, and if I'm not getting like proper nutrients, okay, I am going to be much more susceptible and higher risk if I ended up catching the virus, right? So it's important for me to to jumpstart my immune system right now. So in order to jumpstart my immune system right now, I gotta take the proper diet right now. Maybe I gotta take some broccoli down there, right? Some of the like broccoli, maybe, or what else I could put there? Some maybe garlic there, like some of those, maybe some almonds, and maybe like exercise, for example, like meditations or sort of like yoga, so that way I can boost my immune systems. So it's really important that, to have both physically and also respiratory and also mental hygiene too, to actually boost our immune system. So if we ended up catching the virus, at least we have some sort of immune system that can actually fight for these infections. Okay, so it's really important that, even if you're quarantining yourself, it's really important that you actually take care of your health, with the proper diet, with the proper exercise, okay? And you gotta continue doing that. Anyways, now, after this little talk, we have to talk about, let's say you're one of those people, or like I am, that somehow that we got the exposed to this virus, right? Now, what is this virus is gonna do to our system? That's what we're gonna talk about, it, right? Now, little bit of this viruses right here, this virus has like sort of like this envelope, and these envelopes are made out of like different proteins, the viral proteins. One of the things about viruses is that viruses do not make their own lipid membrane, okay? Virus always steals those lipid membrane from some sort of host cells, all right? So what happens is that these guys right on the black, these are called like S protein, or they simply called spike protein, okay? And this is one of the major proteins we have to talk about, okay? And there is this protein called like E protein too, okay? And these are all glycoprotein. These are M protein right here. All right, let's just name this. And then this one right here on the right point, orange, that is what? That is called my genome, okay? And it's these are coded by this helical nucleocapsid, all right? This is a nucleocapsid. And this spike protein, if you put like in an electron microscope, you put that, this spike protein sort of like looks like a crown, okay? That's why this word corona, the word corona means crown, okay? And that is a Latin word. So that's why we got this word, coronavirus. That's how this virus gets its name, right? Now, what happens is that, okay, so whenever, let's say we take this, we sort of like from the air droplets or aerosols, we take this virus in, right? When the virus comes in, what happens is that it loves to bind us somewhere, and where it's going to bind is that, if I'm going to quickly talk about like some of the concept of here, and let's just quickly make this diagram right here, okay? If I make this one right here, okay, what is this? We're making little bronchi here, okay? Not going to make it this bronchi is primary, primary bronchi gets, these are primary bronchus, right? And we split into secondary, tertiary bronchus, and makes like alveoli and all that kind of stuff, right? And then we'll just quickly talk about sort of like this. Let's just make this as alveoli right here, okay? And then we'll just intertectally, and you have this like all this lungs right here, okay? Now, basically, what happens is that if you want to know, these alveoli cells have that like very two different types of cells, right? So one of those type of cells are called type 1 alveolar cells, or they're also called type 1 pneumocytes. Another one is called type 2 alveolar cells, or they're also called granular pneumocytes. All right, they're called granular pneumocytes, or they're simply called the type 2 pneumocytes, okay? And these type 2 pneumocytes are very, very important for releasing this molecule called surfactants, okay? And the surfactants are what? It's amphipathic molecules, which has both hydrophilic and hydrophobic chains of the amino acids. And the surfactant, what it does is that some of the functions of surfactants is that it reduces the surface tension, okay? It increases the compliancity, okay? And which basically does, it helps with the breathing, okay? It makes the breathing easier, and it has some kind of immunization function too, like it play a role as opsonization, okay? It kind of sort of helps macrophages for the phagocytic processes, okay? That's what it does, okay? And it has some kind of immunology functions also, okay? So it does. And one of the most important surfactants is also act as it has this, right? And this granular pneumocytes, the type 2, it also acts as a stem cells, okay? Which basically means is that if they need to synthesize more of this type 1 alveolar cells or type 1 pneumocytes, because this type pneumocytes are important for like participating gas exchange, okay? It's a participant, very, very important, right? That's one of the main functions of the type 1 alveolar cells. And then this type 2 stem cells, or the type 2 pneumocytes, these will act as stem cells to make more of these alveolar cells. So these are general functions of this, these cells. Now, this type 2 stem cells has abundant amount of this receptor, which this virus loves to bind into it, which are called ACE2. ACE2, okay? This is called angiotensin converting enzyme number 2, okay? I'm going to stress on this, okay? I don't want you to get confused with the ACE2 with the ACE1, okay? And we'll talk about that too, okay? And I'll talk about that too. But what happens is that these guys, ACE2s, are abundantly present in the lungs. They are present in intestines. They're also present in kidneys. But we'll just focus on here on this one. What happens is that, okay, let's just quickly talk about this one, and we'll talk about it. This guy, okay, let's just talk about this in a little detail too. What happens is that you know, guys, like these ACE enzymes are located in the, okay, if I quickly make, let's just make this as, I'll erase this part, but if I quickly make is the lungs right here, right? And what is coming here from is like, if you remember, like a couple of things I have to mention right here. Quickly, not going to go super detailed, but what happens is that the liver always synthesizes this protein called angiotensinogen, okay? And angiotensinogen, okay? And then what happens is that whenever the renin comes in, the renin is actually released by, if you want to remember, like quickly, let's just talk about this too. Okay, what happens is that, okay, this is my kidney, right? And then what happens, okay, let's just make this, okay, let's just make this a little bigger actually. So what happens is that, all right, here, and they have this afferent arterioles coming this side, right? And what happens, this is going to be a glomerulocapillaries, which gets split into efferent arterioles, and this efferent arteriole becomes your peritubular capillaries, right? And this is how it becomes. And then, if you want to become this kidney, let's just make one nephron here, okay? And this is going to be what, let's say Bowman capsule right here, okay? And this is going to be proximal tubular cells here, which is going to make this Henle loop, okay, and then this becomes distal convoluted tubules, and this has become your collecting tube, and then urine will come out here, okay. So now, on the top of this efferent arterioles, they have special type of cells, which are called juxtaglomerular cells, or they're also called granular cells. They're also called pericytes. And then what happens is that, and on down here, this distal part of the early part of distal convoluted cells, they have this special cells which are very dense, which are called macula densa, okay, which they love salt, and they're very, very dark, and they're connected by these cells called Pulchisens, or they're also called, let's just make it, they're also extra mesangial cells, which are because they're outside of the mesangium. That's why I call. And all together, when you compare the juxtaglomerular cells or granular cells with the Pulchisens or extra, or they're also called Lasix cells, and then the macula, when you combine all of this together, we call them juxtaglomerular apparatus. Okay, so these guys are very, very important for synthesizing renin. And renin is an enzyme. What happens whenever the renin comes in? What happens is renin like plucks out some of the amino acids from this angiotensinogens and converts into angiotensinogen number 1. Okay, and this angiotensinogen is an inactive one, and what happens is this will go start circulating, and when it goes to the lungs, and what happens is the lungs has this enzyme. Okay, at the endothelial cells of pulmonary capillary beds, they have this enzyme called ACE1. Okay, and what it does is that it will convert ACE1 into angiotensinogen number 2. Okay, this is a powerful vasoconstrictor, okay? That's a very, very powerful vasoconstriction, and it has a lot of function. It can go to hypothalamus, it can cause increase in thirst centers, so you can drink a lot more water. You know, it has a function in the proximal tubular cells here, okay, to reabsorb your sodiums and chloride. A lot of function it does, okay? But basically what it does is trying to increase the blood pressure, right? That's what the whole functions of this one. Now, and with this angiotensin, what happens is that, like, you know, there is this ACE enzyme, okay? This ACE enzyme, which is ACE2, what happens, it downregulates, it breaks this angiotensinogen and converts into the angiotensinogen like 1/7. And this is a vasodilator, okay? Because it reduces blood pressure, that's what it does. Now, what happens is that that's one of the functions of the ACE2 right here, and which basically, and also all that does it prevents from the lung injury, okay? Prevents from the lung injury. That's what usually these enzymes are doing. Now, if the virus comes and binds into this protein, which is the ACE2 here, what happens? Let's just make this as ACE2 protein right here, okay? This is my ACE2 protein right here, okay? If these proteins are going to be, when a virus binds into it, what happens is that a lot of viruses can come and binding into it. What happens is that the amount of ACE2 enzymes will be reduced. Okay, whenever amount of ACE2 enzymes will reduce, what's going to happen? What is going to go up then? You're going to have an angiotensinogen level would go up, like angiotensinogen 2 level will go up, right? And whenever the angiotensinogen 2 level will go up, there is a role the angiotensinogen 2 will play, because remember, usually it breaks angiotensinogen 2 and make the vasodilator products, right? But whenever if there is already the amount of ACE2, if it's going down, that means what's going to happen, it's no longer break this compound, right? Angiotensinogen number 2. So that means you're going to see increased level of angiotensinogen 2, right? Hold that thought, okay? And we'll come back to that, what is going to happen with angiotensinogen number 2 whenever you have increased level of angiotensinogen or 2, okay? Let me erase this part, okay? But now, come back to this. What is going to happen is that this is my, I'm making these cells as my what? I'm making these cells as my, these are pneumocyte cells, okay, the type 12 cells. So what happens is the virus comes and binds into my ACE2 receptors, okay? So when it's bind to, let's just say this is my virus right here, okay? This is virus right here. So when it binds to this virus and it has genome particles right there, right? It binds into them. What happens is there's also another protein, but now there's lots of bind to this receptor ACE2, right? There's one more protein here, okay? This is, you have to remember this one too, which is called transmembrane, okay? It's a transmembrane. It's a protease enzyme, okay? It's from the serine family, okay? It's called serine family. I think that's what it's called, TMP receptors, okay? So this transmembrane protease serine 2 family receptor, okay? What happened is, okay, so it's transmembrane protease receptor as serine 2 family receptor, okay? It's a transmembrane protease receptor. I could be wrong on that, but this is what it is. It's a transmembrane receptor protease. What it does is that it can cleaves these spike proteins, okay? It cleaves the spike proteins, and then what it does is that when it cleaves the spike proteins, actually it cleaves at two sides. One, it cleaves it, and the other one, it activates it, right? When it activates, basically what this transmembrane protease receptor doing is that it's driving the fusion property, okay? It's driving fusion property. Look, there is a lot of mechanism. There's a lot of process, even though how fusion happens. But let's try to make it as simple as we can, okay? Because, you know, if you're a virologist or if you want to be that, they'll definitely know, okay? Like there's a lot of process it goes into them. And then what happens is that these guys will drive this fusion process, and what happens is that when it starts coming in, it started forming sort of like a hairpin sort of a loop right here, okay? It started forming the hairpin sort of a loop like this, okay? And there is a then after that they will kind of like sort of like release this viral protein down here, okay? And then this viral protein will go uncoating, which basically means they will just, all this envelope will get removed, and only what is going to come inside to the cytoplasm of the cells is going to be this genome, okay? This genome. And this genome, guys, it's crazy. It's gigantic. It's about like 30 kilobase, like 28 to like 32 kilobase, and has like 30,000 nucleotides. It's gigantic, and it has like a 5 to 3 prime. Okay, and they usually call they call this as a, what do they call it, as a positive, okay, single-stranded RNA. Okay, what does that mean? With they're saying plus, there's a plus sense, positive strand RNA, which basically means is that if the ribosomes can come and see it, ribosomes can directly translate into protein. That's what they call the plus sense RNA. Okay, now let's make this. Now this guy's got uncoded and it's in the cytoplasm, right? When it goes cytoplasm, what is going to happen is the ribosome comes in, okay? And the ribosome will start reading it, okay? Let's just make this as that's a ribosomes, okay? This ribosome start reading it now. If this ribosome start reading it, what's going to happen with this is, okay, very, very important things. If the ribosomes start reading it from like both sides, okay? But one of the key factors of this is very, very important. They go something called ribosomal frame shifting. Okay, what does that mean? Okay, ribosomal frame shifting, which basically means is that, okay, they have the opening reading frame, okay, they have opening frame shift reading 1A, that's what they call. And when you start reading it, what happens is that they're going to make some kind of protein, which is called polyprotein 1A, okay? That's what they do. So if I make it, okay, let's just make this, okay, if I make it, they just make this proteins right here, okay, and this is called, let's just say, there's opening frame, they read it and they make this polyprotein 1A, okay? And then at the middle of the time, as it goes from up to here, what happens? They'll shift it, okay? And they'll find another reading frame, which is called like open frame shift 1AB or something. And what happens? They'll start reading it, and they'll start synthesizing another protein, okay? And we'll make this as another protein right here. And this is called polyprotein 1AB. That's what these guys do. And polyproteins are gigantic proteins, right? So basically, with the one, like genomes, like with the one, they're making like lots of different types of proteins, okay? And then after, what is going to happen with these guys is now, this polyprotein, what happens is that there's some of the enzymes going to come and proteolysis happens, like proteolytic happens, okay, and then which basically means is that they'll break down into smaller proteins, okay? They'll break down into smaller proteins, like let's just make it, let's make like a small protein. Let's just make, okay, this is one here. Okay, we'll just make different colors. Okay, this is another one here. Okay. And then we'll make like another one here like this, you know, just making like different colors, and so they'll make like all this kind of proteins, right? Because these are making the smaller like protein particle. That's what happens. And what all these guys do is after that started replicating. Okay, so no, before that, let's just make it this. So this process is like, okay, this process is like, this process is like sort of like a translation, right? Because they're making this to the translations is happening here, right? And they're making this polyprotein, and then after that, what our proteolysis happens, and all these protein particles happens, right? And all this protein particle, what happens? They'll come and kind of like bound with this, like fuse with let's say the 5' RNA, because these are going to go to replication, transcriptase, and all that stuff. Let's just make this. They'll fuse with this guy, which is the 5', 3'. Okay, with our RNA, single stranded RNA, right? Then when they do, then they're going to go to they're going to go something called replication process. Okay, let's just say replication process. Now, when they're replicating, okay, there's a couple of things happens. Okay, when the replications happens, they're going to make two things. Okay, let's just make it in little different things. Okay, let's just make one here, and we'll just make it right here, one here. Okay, and then we'll just write down, okay, what is this going to be? This is, let's say this is 5' here, and this is 3' here, and let's say we'll just 3' and a 5' here. Look, when they're replicated, obviously these guys are going to have two features, right? One is going to be 5' and 3', right? So look, if this guy is like, okay, if this is 5 to 3, look, this has to be 3, and then, okay, what is this? 3 here, and this is what, 3 to 5 to 3 right here. Okay, and this is